our second lab is on paper chromatography. So we have a problem that we need to address. So I have a friend, and my friend is allergic to yellow dye number 5. That's kind of sad, isn't it? So that means that any food that has yellow dye number 5 in it, he can't eat, okay? Here's the thing, there are only 7 different types of dyes that have been approved by the USDA. I put those dyes on the back of your front page of your paper chromatography paper so you can look and see which ones. Now you'll notice if you look on that there are certain colors that aren't on there. There is no green, there is no purple. So how do we get those? Well, it's by a mixture of the others. So not only does yellow have yellow dye number 5, and sometimes yellow doesn't, because some yellows are yellow dye number 40. So we don't even know if this yellow dye is number 5 or number 40. We don't know. So we've got to figure that out too, okay? So just don't assume that they can't have the yellow. They might can if it's yellow dye number 40, right? You might could eat it. So we got to figure that out. So what we're going to do is we're going to see how many different types of M&Ms our friend can eat. If it has yellow dye number 5 in it, he can't eat it. And again, don't just assume that the yellow is number 5. It might be number 40, okay? So we've got to do a problem with that. So to figure that out, we're going to use chromatography. What chromatography is it's a separation technique. There are different types of chromatography. There's TLC, which is thin layer chromatography. There's paper chromatography, which we're doing. There's column chromatography, which you'll do when you get to organic. Lots of different times you'll do TLC and organic too. Paper chromatography is one of the simplest types, but they all do the same thing. They separate mixtures, okay? So what happens is that in chromatography, and this is all types of chromatography, you have 2 phases, a mobile phase and a stationary phase, okay? And how things separate is that some things like the mobile phase better and some things like the stationary phase better. Our stationary phase for our lab is going to be our chromatography paper. So show us our chromatography paper. So this is our chromatography paper. This is going to be our stationary phase. Some of the dyes are going to like this better. Some of the different types of dyes are going to like our mobile phase better. That's going to be the liquid we put in at the end. So when we have that, that's how they get separated, is by how much they like this phase more than this phase. And you'll learn in organic what things like different types of things better. Now, we're going to calculate from that what's called a retention factor. We write that as RF. Now, retention factor is not like density. If I want to know the density of lead, I look it up in a textbook. But if I want to know the RF factor of yellow dye number 5, I can't look it up in a textbook because different mobile phases and different stationary phases have different RF factors. So RF is something to compare with like things. I can't look it up in a book. So that's what we're going to do. Yellow dye number 5 has a chemical name and it's tartrazine. So we're going to use tartrazine as our standard. And we're going to calculate the RF factor for tartrazine. And RF is going to be our distance traveled by the spot over our distance traveled by the solvent, if I can spell traveled right, by our solvent. We're going to find that for tartrazine, our standard, and we're going to compare it to the RF factors of our different separations of our dyes of each color of our plain M&M, okay? So if they have the same RF factor, we can assume that that's going to be that yellow dye number 5, okay? Now, I'm talking about our spots. So what we do when we get our dye, our solution that we're going to use, and we're going to talk about how we're going to get that off of the M&Ms, but we're going to spot these. We do that with capillary tubes. Now, capillary tubes are really tiny, very sharp, so be careful with them. Glass tubings, okay? And you're going to take and dip it in your solution, and then you touch it to your chromatography paper. Couple of things about your spots. You don't want your spots really, really big, because if you have 2 spots that are really big, when they start to separate out, they will interfere with one another. So we don't want them really big. If you do them too small, you're not going to be able to see them. So you want to do them to where they're concentrated. So what you're going to want to do is you're going to want to spot it, kind of let it dry, wave it around, put it under the heat lamp. We've got hair dryers you can use. Then spot it again. Let that dry. You want to do that 4 times to get it concentrated. Okay? All right. So we're going to dot those, and to practice, because our chromatography paper is pretty expensive, so we don't want to mess that up. That's right, you don't want to mess that up. You're going to get one sheet, you don't want to mess it up. We're going to put some filter paper out there, which isn't as expensive, for you to practice dotting on, so that you can practice it before you just get to your piece of chromatography paper. Okay? When we write on the chromatography paper, notice that I've put the colors of my M&Ms, red, orange, yellow, blue, green, brown, and then a T for my tartrazine. We want to write on this only with pencil, because if you write on it with pens are made up of different colored dyes, so when you put it on there, it's going to go right with our other colors, so it won't interfere with it. When we calculate our RF factor, so you're going to get spots. So let's say our tartrazine spot looks like this. Just kind of measure to the middle of that spot. So you're going to measure for the distance traveled by the spot from where you dotted it all the way up to about the middle of that spot. For the solvent, once you're done, you're going to get the paper out. It's going to be wet. You want to take a pencil and mark across the top that solvent front. You need to do that because once it dries, you won't be able to see where that is. So what you want to do is mark across that solvent. And then once it dries, you want to get the distance from here up to the solvent. That is this portion here, okay? So don't measure from the bottom, you measure from here, okay? All right, so that's how we end up calculating our RF factor. Now, how do we get these solutions? Okay, well, when we have an M&M, okay, you might say, why don't we just get the color off the M&M and use that? All right, Paxton, is M&M, is it just have dyes in it? No, what else is in an M&M? Chocolate, sugar, stuff like that. Anything like that's going to mess up our chromatography. So we want just the color. So what we're going to do is we're going to use yarn, show them our pieces of yarn. They're really, really small. All right, but what we're going to do is we're going to take the dye off the M&M, and then we're going to dye this piece of yarn so that it is the color of our M&M. Then we'll remove the dye from the yarn to make our solutions. So that way we don't get the sugar and the chocolate and the fillers and all that kind of stuff. Now, the first thing we do though is yarns have kind of fluorescent dyes on them. So even like white yarn does still have some dyes on it. So what we're going to do is we're going to boil these to get any of that off, because again, any dye on the yarn would affect our chromatography. So we boil these first. Now, show them how that we use our test tube holders with our test tubes. So when you're boiling something with a test tube, you don't want to stick your hand in there and grab the test tube. So show them, these are our test tube holders. So how do you open them? How do you open them?
Squeeze on the little holes and just squeeze it out.
And then you put the test tube in. And once we get it on there, we can let go of the test tube, but what do we not do?
We don't squeeze the holes in it because it can come out of grit.
And then we've got to start the experiment all over again, and we don't want to have to do that because that's more of our time, right? All right, so be very careful you don't squeeze the test tube holder again once that you've got it there with the test tube. Okay, so we're going to boil our yarn, and it says to pull them out with tweezers, but show them a problem with our tweezers. Yeah, we put our yarn down, and there you go. So what happens?
It doesn't go all the way down.
It doesn't go all the way to the bottom. That's right. So what I suggest is that we get a waste beaker. That's right. And that we do what? We pour our solution into the waste beaker, and when we do that, the yarn will kind of come down. If it doesn't, we very lightly kind of tap the test tube onto a paper towel, and the yarn will either come out or it will come far enough down that you can pull it out with the tweezers. Okay? All right, so once you're done with that first waste beaker of acetic acid that you got the dye off with, you can put that down the sink, no problem. Once we get our yarn ready, now we're ready to get our M&Ms. So we're going to take an M&M and we're going to put it down into our test tube. You can use those same test tubes because it's just had acetic acid in it, so it's the same thing. Now, Once we get our M&Ms, and you're going to have 6 test tubes, each with each color in them. Once we get them in there, we want to boil them only long enough to get the candy coating off. And you'll notice it coming off. You need to really watch and pay attention because we want as little kind of chocolate and that kind of stuff in it as possible. So once the candy coating come off, the only one that's hard to tell is the brown one. That's right, the brown one's kind of hard because it's why. Why is it hard?
It's brown.
And it's the same color as what?
Chocolate.
The chocolate, so it's kind of hard to tell. The rest of them, it's pretty simple. So as soon as the candy coating comes off, pull it off, and then again, get your waste beaker. Oh no, we're not going to use the waste beaker there. What you're going to do is you're going to have a second set of test tubes. You want to pour that solution that has the dye and it has the sugar from the candy coating and maybe a little bit of chocolate. You want to pour it into a second set of test tubes. The M&M, please, for goodness, what do we not want to do with that M&M that's in that test tube? Eat it because it's been in acetic acid. So just toss it into the trash, okay? So you've got your solution with your different dyes in it. Now you're going to put your yarn in there because now we're going to dye our yarn so that we only get the dye and not the sugar. Again, it tells you about boiling it again, tells you what to put in there. Now, you need to be sure that the yarn stays in the solution. If you need to get a stirring rod and kind of push it down, Ms. Carmen and I, when we did this lab, we did not have trouble keeping the yarn down in the solution. But if you do, you need to take your stirring rod and kind of push it down there. Now, be careful. If you do that, let's say that you do that in the green one, you've got to rinse your stirring rod off with distilled water before you put it in another, because if you don't, you're contaminating. And you're going to see those colors elsewhere. So be very careful with that, okay? So then you're going to dye your yarn. It tells you about how to do that. Once that you've boiled it the length of time it tells you, then what you're going to do is you're going to pour off that solution, okay? Now, again, pour it into a waste beaker, pull your yarn out, okay? Now we have to take that dyed yarn and remove that from there, okay? Now to release that, you're going to, and this is very, very important, you're only going to add enough ammonia to cover the yarn. You need to get a concentrated dye solution. Ms. Carmen and I did this lab lots and lots and lots of times so that we could get this just right. And one thing that we found is if you add too much ammonia, it's not concentrated enough, and then you have to boil the solution and you can't see the color. So be sure that you don't add too much ammonia. It tells you to use just enough to cover it. That's usually about a milliliter. So kind of push the yarn down in the bottom of the test tube so that you don't have to put a lot in there. Again, heat it for 5 minutes and to pull that back off, okay? After that, remove the test tubes. You're going to put those in your test tube rack. At that time, you've got your solution that you can dot your chromatography paper with. Now, I've already talked about dotting the chromatography paper, but one other thing I want to talk about, once you get it into the solvent, and it tells you your mobile phase, your solvent is 1% sodium chloride solution. One of the things that you need to be careful of is this. Once you put your solvent into the test tube, you've got to be sure that the solvent line doesn't come above where your spots are. That's another reason why you don't want to do a really big dot. If I do a really big dot like this, and I pour my solvent, and my solvent is already right here, then this dye is going to come off in my solvent, and I'm going to see it all across my chromatography paper. So be sure that your solvent in the beaker is below your dot line. Okay, that's very, very, very important, or else you're going to have a big, big problem. Okay, so what you want to do is you're going to take and you're going to get your chromatography paper, you're going to fold it around, and you're going to staple it. Be sure that the two ends don't overlap. That's also very important, okay? All right, you're going to let it go until it gets close to the top. It says about 1.5 centimeters from the top. That's going to take a little while. When you're waiting on it, we don't want the solvent to evaporate, so we put parafilm over the top of it, and we'll show you how to use the parafilm when you get into the lab. Once you pull this out, you've got the solvent about there. You're going to let it dry. You can do that with a hair dryer under the heat lamp. and then you can calculate your RF factors, and then you can answer those questions. So you're going to show all of the RF calculations, and then you're going to answer that question about which ones that your friend cannot eat and which one they can eat. Again, tartrazine being our standard, it is our yellow dye number 5. Okay? All right, so that is our paper chromatography lab. Just be sure that you're really careful, that you're not pouring things, contaminating things. That's very, very important with this lab, because if you contaminate, you're going to see a lot of these all the way across. Again, you need a good, nice, concentrated dot, so that's very important at the end, not putting too much stuff, and also being sure you get a concentrated dot there. No, we're good. All right, so that's it for chromatography. We'll see you next time.